Okay, once again we are back with the mammalian histology of that trachea, histological structure of the trachea respiratory system. In the previous lecture, we have seen the detailed histology of trachea. Now we'll go advancing towards the main bronchus or bronchioles, and we'll see histology of bronchioles now. Now here, just to revise the windpipes, all this duct system internally lined with the epithelial tissue. Then they are embedded with these epithelial tissue are embedded with the goblet cells. They are ciliated. All these epithelial tissues are ciliated. Then in submucosa portion they have these glands, and they are internally supported with the hyaline cartilage, and there is a smooth muscles present in a in a adventitia layer as well as between the hyaline cartilage ring, and they are all entirely so these elastic fibers in a connective tissue are present. So overall, there is a little variations as we go from the trachea to the terminal bronchus level. Now here in this diagram, they have tried to show how this type of epithelium, number of goblet cells, ciliated cells, glands, hyaline cartilage, smooth muscles, injective fibers goes on varying depending on the area where that windpipe histology is there. Okay, so in trachea you will see all these component present in a good quantity, but when it reaches to the main bronchus, that is the two branches level of this windpipe, you will find that goblet cells are enlarged, and the columnar epithelial cell size goes on reducing, and here hyaline cartilage ring goes on reducing in a bronchus, that primary bronchus level. Rest, smooth muscle, elastic fiber are everything is there. But again, when that primary bronchus bifurcate into the secondary bronchus level, in that the goblet cells goes on reducing. Number of goblet cells reducing, column size is reduced, and cilia also goes on reducing. Here goblet cells indicate that they are reduced here. Hyaline cartilage very sparingly available there, and elastic fibers are in good quantity, and smooth muscles are also there. Ciliated cells are there. But when they reach to this terminal bronchus portions, see how that columnar size of epithelial tissues reduce, and entirely in the terminal bronchus there is no goblet cells are present, no glands are present, hyaline cartilage is totally absent, and smooth muscle number is reduced, but elastic fiber remain as it is. And at this bronchus level, that is above the alveolar level, you will find that epithelial type differs, it changes from columnar to the cuboidal one, and there is totally absence of the ciliated cells, glands, no mucous cells, hyaline cartilage vanish, and in alveolar portions, alveolar duct and alveolar sac, alveolar portion, you'll find that there is a simple epithelial layer is there, and nothing else is embedded in that epithelial layer. Very small amount of smooth muscles and very small amount of elastic fiber you see in this alveolus portion. So this diagram indicate entire histology of a tracheal system, that windpipe system. Now here conducting portions. Now from bronchi, from branching of trachea from bronchi to the alveolus, that portion is called as a lower airway, upper airway or upper portion of respiratory system, and the lower portion of the system. So here from primary bronchi to alveoli, that system is called as a lower airway portions or conducting portions of a respiratory system. It is also called as the main bronchi or primary bronchi or extrapulmonary bronchi. Now histological structure is much more similar like that trachea, and they are anatomically in human. That right side is wide and more vertical than the left. But we are learning mammalian histology, so that anatomical structure of the human respiratory system is not related with our present syllabus. Here, as already we discussed with that image in first slide, this histology of secondary and tertiary bronchi is discussed here. The mucosa with respiratory epithelium, and the structure is much more similar like the trachea and primary bronchi. Smooth muscles are present, submucosa and mucosa. In between the submucosa and mucosa, you find that that smooth muscles. They are in all these smooth muscles are innervated by the sympathetic and parasympathetic system. So you may have taken some time these medications for the cupping. So that time these medicines majorly act on our sympathetic parasympathetic system to reduce the cupping response. So we feel dizziness, and they have a label on that bottle of a cough syrup that it may cause dizziness, to avoid driving and like that thing. So this is the relation of that medicine with how respiratory system is related, and these windpipes are innervated with this nerves, sympathetic parasympathetic nerve system, which initiate the cupping response in our respiratory system when needed. Thereafter, there is a submucosa with the seromucous glands, hyaline cartilage plates are there, not a complete ring is there, and the regular adventitia is as it is. Now here is a photograph of a bronchus, secondary bronchus, that is the branch after the trachea. You will see very small fragments of a hyaline cartilage are visible. The rest of the thing is same, like that epithelium layer, submucosa, then this muscles portion is there. And now slowly after the trachea, these smooth muscles, instead of remaining between the two end of the C-shaped cartilage, here that smooth muscle spreads behind that submucosa. After submucosa and adventitia, you will find there is a band of running smooth muscles. Here again the division of histology structure of bronchioles. The mucosa is lined with the respiratory epithelium with sclera cells. Now goblet cells, which are all previous, they are present in the tracheal portion to produce a mucous cell. Now these goblet cells are replaced with the sclera cells. Now these are the dome-shaped cell without any cilia or secretions. They secrete glycosaminoglycans, and they are the secretory proteins. These sclera cells increase the bronchioles and gives rise to the terminal bronchioles. As you go on advancing, sclera cell number goes on increasing. The epithelial gradually become mostly sclera cells with cuboidal rather than the ciliated epithelium as the terminal bronchus near the respiratory bronchi. Already we discussed this thing. The structures of epithelial layer goes on changing. Slowly the cilia vanishes, and that is replaced with these sclera cells and the cuboidal cells. There lies a smooth muscles layer below the after the mucosa. You will find there is a smooth muscle layers, and the cartilage is absolutely absent and adventitia as it is. Now here in this electro micrograph, in this electromicrograph, you will see a presence very less number of ciliated cells, and gradually sclera cells are increasing. Here you can see the sclera cells are increasing. The role of sclera cell is secretion of some proteins, glycoproteins, responsible for the functioning and releasing the surface tensions over the mucous in lungs, alveolar systems. Along with this epithelial, ciliated epithelial and cuboidal cells, there increases number of sclera cells. And rest this portion is submucosa portions. This is a photograph of bronchioles. When you take cross section through the lungs, it shows many more things rather than particularly a bronchi. So here all these structures are alveolus. Small alveoli are cut into sections. Then these ring structures, these are the terminal bronchi or bronchioles. Here, this is a bronchioles. So in one section only, you will see lot many things together. And here you can see there is the absence of cartilage, and only the epithelial layer you can see clearly, and the adventitia portions and surrounding membrane which demark that alveolus from the bronchioles. This photograph of slide of histological slide of that bronchioles, terminal bronchioles, that is the one which lead directly into the alveolus. This is stained with the hematoxylin and using this violet color are the nucleus, and the hematoxylin stain responsible for coloration to this violet color nucleus, and all that pink color portion is the cytoplasm. So here you see that cuboidal cells are more, and no ciliated layer is there on these epithelial tissues. Then this pink color portion is a smooth muscles, and in between there is a this mucosa portions surrounding these submucosa portions and the adventitia portion here. Respiratory portions, these bronchioles tubes, that respiratory bronchus means the tubes between the alveoli. An alveolar duct or alveolar sac arises from respiratory bronchus but have more alveoli and terminate as blind pouches. Now, these blind pouches are what we say alveoli. Alveolar three-dimensional structure, you will see that this is a grape-like, single grape-like structure attached with the stem. That stem is a bronchiole which goes on collecting and forms the tertiary bronchi, secondary bronchi, primary bronchi, and trachea. So the end of a respiratory windpipe is a alveoli. Alveoli, they are thin-walled pouches lined by type 1 and type 2 pneumocytes. Now this pneumo word is related with the air, and where the air exchange take place. So that cell are named as a pneumocyte. Type 1, type 2 pneumocytes are there. This is the actual site where gas exchange take place. Now this gas exchange thing not only carried out in a alveolar duct, but it start from the tertiary bronchi. So gases exchange take place. It's initiated from the tertiary bronchi. Whatever the internal lining, that epithelial lining is there on the tertiary bronchi, from that surface only the gases exchange take place, and it goes on increasing as we advance to the alveoli. They are lined by cuboidal cells and connect to the alveoli and alveolar ducts. Here in this photograph, you can see presence of respiratory bronchi, alveolar ducts, and alveoli. Now you have to little bit train your eyes to get understood this one unidirectional photograph, because when we think and we learn that histology in three-dimensional fashion, but it don't look like what is there in a theoretical three-dimensional descriptions. Here, when we see observe that slide under microscope, it is we have to make correlate what we see with what we learn. So here, see, go on following these labels, so that will help us to understand how this slide should be read. So here, this respiratory bronchiole are there, and these are the alveolus in the wall of that bronchioles. This most of the wall of the bronchioles has a definite line of dark along with it. You see this dark line. So because of this dark line, we are able to identify it as a bronchiole. That signifies the cuboidal epithelium. When we say that there is a cuboidal epithelium, because of this dark line color, we can understand that there is a cuboidal epithelium, and so it is a respiratory bronchioles. And now this portion you see here, and this portion here, and this portion here. These are the many alveoli opening in central portion. Okay, these all alveoli they open centrally, and they form this alveolar duct. Its wall consists almost entirely of alveoli, which have only a simple squamous lining, too flat to be visible there. Now, you don't see here cuboidal cells, epithelial cells. They are simple squamous epithelial lining. Then these round structures, they are alveoli, and these are the, I think we have discussed earlier, blood vessel, histology of blood vessel. So if you carefully observe these portions, you will find centrally there is a RBCs and this endothelial lining and all that RBCs, all that blood vessel histology you can see here. So this is a blood vessel, pulmonary blood vessels, which provide or they exchange blood through these alveolar portions. Okay. So in this entire photograph of the actual lungs cross sections, how to read it? All these vacuole-like portions, they represent the alveoli. Same when you see lot many alveoli opening centrally, that portion is called as a alveolar duct, and these all alveolar duct combine together and open into this bronchi. Now, why this portion is a bronchioles? Because it is lined with the this cuboidal epithelial cells, and they are dark in color, and all these are lined with the simple squamous epithelial line. This help you in identifying a slide in a practical courses. When we keep that slide, lung slides under microscope, and we have to identify, there is no label. By seeing such structures, you can correlate that this is a lungs TS. Again here in another photograph. Now I think you can easily identify the structure here. See this dark line portion, some showing some cuboidal epithelial layer, and this pinkish color showing the muscles, smooth muscle layer, adventitial layer. So this must be a bronchiole. Okay, and this is central portion is a mucus accumulated here and stained. So this is a bronchioles. Now in this photograph, these are the bronchioles. These round structures are the bronchioles. Okay, these are the blood vessels, or that may be some bronchioles cut into sections. This is a vertical cut of a bronchiole. They are the vertical cuts of a bronchiole. Now, what about this portion? See, you can see the structure. Cellular structure is simple squamous epithelial layer, and these are the all round alveoli cut into the different plane and forming this alveolar duct, and these are the alveoli now here. So here this portion is a alveolar duct, and this is the alveoli. These all are the alveoli. Generally during processing of this slide, during micro technique, many times these lung alveoli get collapse. If not impregnated properly with the wax, they get collapse, and then it is very difficult to identify that slide as a slide of a trace of a lung. You have to scan many slides of same block, same tissue, then sometime somewhere you get the proper architecture design of this architecture design of this arrangement of tissues, and you can able to identify